Welcome to Unfreeze Your Science, a podcast powered by CryoPDP, a temperature-controlled logistics company for the life sciences and healthcare industries. And now, it's time to unfreeze.
My name is Joris Deelen. I'm a research group leader at the Max Planck Institute for Biology of Aging, and I work there on human aging and specifically on the genetics of human aging and biomarkers of aging, which I want to share you a bit about today. So, as you probably know, we are getting older and older. So the population, the lifespan in the population has been increasing steadily over the last 200, 300 years. And now we are reaching kind of a phase where it starts maybe to decline due to some bad influences that we are having, but overall it's still increasing in the world. And sadly, this increase in lifespan has not been accompanied by a similar increase in health span. And what's then health span? So health span is the time that you bring your life into good health. So how long you live in good health. And sadly, although we have been extending our lifespan, our health span has not increased at the same rate, which actually means that at the end of life, people are getting more and more diseased. And this is a problem because we know that actually aging is a major risk factor for many age-related diseases in general. So, for example, diabetes, cardiovascular disease, also some forms of cancer. So although we're getting older, we're also getting more sick. And actually, the goal of my research is to find out how we can actually extend this health span. So we're not so much interested in extending the lifespan even further, but really trying to increase this time spent without the occurrence of chronic age-related diseases. And when I was still a master's student, I became interested in this topic because I came into contact with a group which was actually looking at these very long-lived individuals. So individuals that were above 90 years of age at the time of inclusion, which was 20 years ago, so they were really old. And then they also had multiple family members which were also this old. So they really have something special. So these families together became very old, potentially because of genetic factors, but it could also be because of shared environmental factors. We actually think the genetic component is a bit stronger there, but it might also be that they have certain ways of living that make these families longer lived. And so I came in contact with my former supervisor, which was Eileen Slagboom, who collected all these families, and she was looking for somebody that would look into the genetics of those people. So can we find something in the DNA that make these people so long-lived? And if so, how does that work in the general population? Does the general population also carry this, or is this something unique for these long-lived people? And very interestingly, if you look at these long-lived people in general, they're also a very special part of the population because they actually do not only live longer, but they also live healthier for longer. So they could be a good natural model for increased health span as well. So then I started my PhD on this subject, and the first thing that we did is looked, okay, can we find something that is shared between all these people in their DNA that can explain why they are so long-lived? So we took these long-lived people and these family members, looked if there were specific variations in the DNA that were only present or were actually more enriched in these long-lived people in compared to normal-lived people, so the so-called controls, and then look, okay, can that explain why these people live so long? But actually, we were not so successful with this approach because it doesn't seem to be that these long-lived people carry something that they all have in common that can explain why they are so long-lived. Instead, we actually think that it's probably a different kind of mechanism by which each family has a different mechanism or different way by which they became so long-lived. So we cannot find something in common, but probably unique to each family or to a couple of families only. And this is actually further supported by evidence that we gathered afterwards, because then we not only looked into this long-lived population that I just talked about, but actually collected long-lived people all around the world and did the same kind of analysis. So looking if there was something in their DNA that was shared between these long-lived people and enriched in comparison to controls. Also there, not much was found. The only thing that we actually found, which is maybe interesting to mention, is this ApoE gene. So in this ApoE gene, there are two different kind of genetic variants, the ApoE4 variant and the ApoE2 variant. And the ApoE4 variant seems to be depleted in the long-lived people, while the E2 variant seems to be increased in the long-lived people. And this is actually quite interesting because these specific kind of genetic variations had already been coupled to specific diseases. So the E4 allele, people that carry it have an increased risk of Alzheimer's disease and cardiovascular disease, for example. And the people carrying the E2 allele have the opposite, have a decreased risk. And we actually can also see that then in the long-lived people, that of course we expect that the long-lived people would have less of such a disease risk allele and more of a protective allele. And this is actually what we're seeing. But it's kind of the only thing we pick up in these large studies. So that was for me a reason to shift gears a bit and no longer look at this shared variance across many different people, but really start looking at very specific variants. But that, of course, also brings in some risk, because how do you study such variants that are unique for long-lived people, right? How can you actually show that such a variant is causally related to the lifespan of these individuals? So I can then up with an approach where we first focus on variants in genes that are involved in specific mechanisms where it's already known that they are contributing to lifespan regulation in model organisms. So if you take worms or flies or mice, there has been quite some studies which show that there are specific pathways that are conserved between species that extend lifespan. And one of these pathways is the insulin signaling pathway. So we also think and we hope that there's also in humans some variation in this pathway that can lead to a longer life. And this was the starting point. So we start with this pathway and then look, are there specific genetic variants in this pathway that are seen in this long-lived people and not seen in the general population? And then we were able to find quite some variation. And then the next step is, okay, how do we continue from there? So first thing we did is restricted ourselves to really variants where we have evidence, at least bioinformatic evidence, that they really influence the functioning of the protein. So they are in a region of the protein where actually, if you expect that you make a mutation, it will potentially lead to an effect on the protein itself. And like I said, they had to be really unique to the long-lived people, so have not been seen before. And when we continue then with around 10 variants, the next step is, okay, how do we follow this up? So what we decided to do is we decided to make cell lines containing this variant. And for this, we used the CRISPR-Cas9 system, which you might know as the cutting and pasting of the genome. So what we did is we introduced these specific mutations into cell lines. And then we looked into the cell lines, what happens with the protein in which these mutations are present and also with the downstream mechanisms. And actually, very interesting, we see that the far majority of the mutations that we selected do something at the level of the protein and also at the pathway. And they seem to kind of cluster in two different groups, which each seem to be potentially health promoting, but how they exactly work, we are still studying. And as a next step, we also thought, okay, what can we do on top of that? On top of the things that we can do in the cells, how can we provide more evidence that they can be causally linked to the lifespan of these long-lived people? So then we decided to also start generating some mouse lines, also with CRISPR. So we took again this variants of interest and we brought them directly into mice. So then you have these mice that are normal mice, except for one specific genetic variation that they got from the humans. And then we look, okay, first of all, do we see an extension of the lifespan of these mice, right? Because that's what we are hoping for, that if they come from this long-lived people, that also in mice they would extend lifespan. It's not necessary, it's not a requirement to show that they are functional, but it, of course, can show some evidence that they could be linked to that. So this is one thing that we are checking. And this research is now ongoing for about 2 years. So we start the first lifespan experiments are kind of now in the phase where we can start seeing potential differences, but it's still, I would say, 1 or 2 years before we can actually say that they do something with lifespan. And the second thing we do is also we look at the phenotypes of these mice. So we do extensive phenotyping of the mice, where we actually look for example, how powerful they are with grip strength, how well they can move, their balance, also kind of how their insulin signaling is behaving. So you can do this test, so that they're also used for people to determine if they have diabetes, which are so-called glucose tolerance or insulin tolerance tests. You can also do in the mice, so we also do them there to see if they are altered. And we just look at kind of general behavior of these mice, with the idea that hopefully these genetic variants that we bring in lead to an improvement of these phenotypes, so that they actually are mice are potentially more healthy than you would expect from the same mice from the same age that do not have this kind of mutations. And the third thing that we are doing is also collecting tissues from the mice. So we have different cohorts where we at different time points collect tissues to see what's happening there, and that we can hopefully also translate back to the cells. So if we have some of these variants that show then an interesting effect, then of course the question is what to do next, how to continue from there. So if you know that a specific variant leads to a healthier life, what can we do to benefit the general population? So this is in general the question. What can you do with genetics that can benefit the general population? So then also the idea is that what we want to do is go back to the cell lines that we have created, where we saw also these effects of these mutations, and then try to treat these cell lines with different kind of drugs. So different kind of medications that are, for example, already used in the clinic, but also kind of novel medications that are still being designed. And then see if you treat these cells with this type of medications, does that show the same effect as this mutation? So can specific drugs mimic what we are actually seeing with the genetics? And if that's indeed the case, then we can, of course, translate that back to the humans and say, okay, we think that these types of drugs influence these specific mechanisms which we are known to be altered in these long-lived people genetically. Can this also help to improve healthy aging in the general population? This last part is still a couple of years away, I would say. I mean, we will start now seeing if we can mimic these effects, but to go from there all the way to developing something to use in the clinic takes a long time. So we are just starting with that. So this is kind of in short the main research that is going on in my group, and I know there are also other groups in the world that kind of start using similar approaches, because this common genetic variation, so the variation where it's more prevalent in the general population, does not seem to be successful for longevity and aging. For other traits, it's very successful. If you take, for example, cardiovascular disease, and you do these large studies, you identify many different genetic variations that influence this trait, which you can then follow up. But for longevity, it seems to be more complex. And one of the reasons is also that we think that maybe 10 to 15% of how long you live is genetically determined. So the far majority is still environmentally driven. And this is also kind of where my second part of my research group is working on a bit more, because we have this genetics and we can come up with things like that, but also we want to know more in general, how can we actually determine if somebody is healthy or not in the general population? So instead of using genetics, what kind of other factors can you use? And there comes in the so-called biomarkers. So the idea of the biomarker research is to identify, are there specific traits or substances in your blood, for example, that can predict how healthy you are. And this is a big line of research in the field, because, of course, what we want to do is we want to identify people that are on track to become unhealthy and develop diseases, but we want to intervene before the diseases occur. And this is with aging, we expect that if we target aging, we can prevent many different diseases at once that all happen at the end of life. So the idea is, what we want to do with these biomarkers, we identify people a little bit early in life, say 50, 45, before they actually show the symptoms of sickness, so that we can start treating them on time. And how this research is working is that we, for example, take blood samples from individuals when they are 50 years of age. We follow those people for a long time, and then we actually see what happens to those people. And then we go back to these blood samples and look into the blood. Okay, what were specific substances in the blood that were indicative of what would happen to these people? And you can do that at different levels. So you can do that with just general things that are already done in the clinic. So you can take somebody's glucose level, for example, or cholesterol level. It can work like that. But you can also do more specific kind of measurements. And a very popular thing at the moment are these epigenetic clocks, as they are called. So what they are doing is they measure the so-called methylation and then look at this as a predictor of how long you live or how old you are. And they say, okay, if we measure this epigenetic profile from you, we can say you are potentially biologically younger or older than your chronological age, or your actual age that is in your passport. And we did something similar. So, but instead of using the epigenetics, we used the so-called metabolomics. And metabolomics is kind of an outcome that you can quite easily quantify in the blood, and is kind of all kind of different substances that play a role in important processes in your body. And this again includes things like your glucose level, also your cholesterol levels, also things like amino acids, so the building blocks of proteins in your body. And there are many different types of substances. And when we did this, actually, we could see that if we take a subclass of 14 of these metabolites, it's very well predictive of how long somebody can live. So we take those substances when the people were younger, we look how long they lived, and then we got back and said, okay, these kind of markers are very predictive. And again, these include things like glucose and cholesterol, which is no surprise, because we know that, of course, higher levels of these substances lead to specific age-related diseases. But the thing that we show new is that if you measure them all in combination, it is very predictive. And then the next step is, okay, how can we use this information again? So what we want to do next is then see, okay, we know that these specific substances are predictive of how long you live. How predictive are they about your current health? So how healthy you currently are. And then you can associate it, for example, with specific diseases, or also specific outcomes like how strong you are again, how fit. And actually, it turns out that these factors that we identify also work quite well on those outcomes. So the next step for us is now, first of all, to improve it and measure it in more studies and see if it holds. But second, also to bring this already a bit more into the clinical setting. So the measurements that we have been doing are more epidemiological based, as they call it. So you take big studies from a population where these people were followed up, and then you go back to the freezers and take their blood. But we now want to see, okay, can it also work directly in the clinic, where somebody comes into the clinic, you measure these substances in their blood, and then based on that you can say, okay, you're now so and so healthy or unhealthy, or more healthy than you would expect. And the people that are then unhealthy, we can already start treating them and see if we can improve the profile over time, because that's of course the goal. We don't want a marker of, say, how long you live or how healthy you are that you cannot change. The ideal situation is that you have a marker which we know you can still change by, for example, lifestyle, or nutrition, or if needed, pharmacological treatment, so that you can kind of bring this profile back to normal and in that way prevent some things to happen. So this is kind of the next step in our research where I'm working on. And this is also a very hot topic in the field, where many people are trying to identify such biomarkers of aging. So like I said, we did it with so-called metabolomics, people did it with epigenetics, so this methylation, but you can also do it with all kind of different approaches, not only based on markers in the blood, but you can also base it on more physical related traits. So for example, a good predictor of mortality is also how much exercise you have, right? So the less exercise, the more increased risk you have of dying. So all these kind of factors are currently taken into account, and ideally we want to start combining all this research and combining all these different kind of measurements to get a good indication of which people are more healthy and which people are less healthy and should thus be treated accordingly. So kind of these are the two things I'm doing in my group, and Like I said, the majority of the work is on the genetics, and hopefully in say 1 or 2 years, we could see that this approach works, that we can actually have this genetic variance where it's shown that they can potentially improve health span and lifespan in mice, and then go into the next phase where we bring it back to the humans. And at the same time, with the biomarker research, we already have a starting point, and now we're starting to implement it into the clinic and see, can we really use this kind of markers to improve clinical decision making in the population. So if you're interested, please visit my website, get in contact, and we can discuss more if you want. And thank you for your attention.
Thank you for listening. We hope you've enjoyed this episode. Stay tuned and discover more about the work of our guest in the description. Podcast powered by CryoPDP, a temperature-controlled logistics company for the life sciences and healthcare industries.